In 1958, Meselson and Stahl created an experiment to test the major hypotheses for DNA replication. In the hypothesis of conservative replication, an entire double-stranded DNA molecule is a template for a new molecule of DNA. The original DNA molecule is conserved. Another hypothesis is the dispersive model, which states that the two strands of a DNA molecule break down into fragments and serve as a template for new DNA fragments. The original and new fragments reform into two DNA molecules. The final model is the Semiconservative Replication hypothesis. The two strands of a DNA molecule separate and each become a template for a new strand, forming two DNA molecules, each with one new and one original strand. In the experiment, Meselson and Stahl grew E. coli cells in 15N, which is a heavy isotope of nitrogen. They took a sample of the DNA and used equilibrium density gradient centrifugation to distinguish the nitrogen. Only the heavy nitrogen was present. Next, they added 14N, which is a normal nitrogen, to the E. coli and allowed them to replicate. When the next generation was tested, it was found that there was an intermediate band that contained both heavy and normal nitrogen. This disproves the conservative model because the original band of heavy nitrogen was no longer present. The cells continued to replicate, and after each generation, the nitrogen in the DNA was distinguished through centrifugation. In the third generation, there was both an intermediate band and a band for the normal nitrogen, or the 14N. All of the following generations had the same results, but with an increasing amount of 14N in each generation. This disproved the dispersive replication model because with this hypothesis, there would have only been an intermediate band that contained an equal amount of the two different nitrogens. This observation helped Meselson and Stahl show that DNA is replicated through the semi-conservative model. With each generation, there is more new DNA, but some of the original DNA is still present.